Hey guys, so let's talk about meiosis. So in section 2, we're going to talk about meiosis, which is similar to mitosis. So let's review the cell cycle real quick. So first off, we start in the G1 where we're growing, and then we get to the DNA synthesis phase, and we synthesize all of it. And then G2, we start growing some more. And then we go through mitosis and cytokinesis, and boom, two daughter cells that are genetically identical that will start the G1 and back over again. So, what is meiosis? Meiosis is when we go through this process of forming daughter cells that have half of the chromosomes that the parents have, or haploid cells. During mitosis, we form diploid cells, which are identical to the parent. But in meiosis, we have half of the genetic information, and those cells are haploid. Those are known as your sex cells or germ cells. They'll produce eggs for females, sperm for males. During meiosis, we start with a diploid cell, and it goes through two divisions to form four haploid cells. So we have meiosis 1 and meiosis 2. In meiosis 1, we have homologous chromosomes that are separated, and in meiosis 2, they have homologous chromosomes that are separated to form four haploid cells, four sperm, three polar bodies, one egg in females. And so what do we have to demystify here? So let's start looking at what we got. So we start with 46 chromosomes in interphase and 46 chromatids. By after interphase, we have gone through the S phase, and we now have 92 chromatids and 46 chromosomes. After meiosis 1, we have 23 chromosomes, 46 chromatids. And then after meiosis 2, we have 23 and 23. Notice our chromosomes with one chromatid are like little butterflies, and then chromosomes with two chromatids look like our X's here. So notice we start and we have X's, and notice we have just the butterflies down here. And so this is what we're going through. We have 1, and then going to 2, and then we end up with 4. Notice the 4 at the bottom are completely different. Well, that's a reason for that. You don't look like your siblings, do you? So let's talk more about that. So again, the stages of meiosis are similar to mitosis, such as we'll go through PMAT, prophase, metaphase, anaphase, telophase, and then there's cytokinesis, and we restart it over to prophase 2. Okay, so let's look and compare prophase 1 versus prophase 2. So in prophase 1, you'll notice that the chromosomes are condensed and the nuclear envelope breaks apart. That is the same thing that happens in mitosis. But in prophase 2, new spindle fibers form around the chromosomes. So now we don't just have one set of spindle fibers, but we have two sets of spindle fibers. In metaphase 1, you will see your homologous chromosomes will move to the cell's equator. Notice we have chromosomes that are lining up there. They're homologous, which means there's two pairs of the same. So you have two number 1s, two number 2s, two number 3s of all your chromosomes, 1 through 22 will all be lined up there. And then you have your X and your Y. There's the sex chromosomes. And then in metaphase 2, the chromosomes still are lining up on the equator, but notice they're not homologous pairs anymore. In anaphase 1, we will have these homologous pairs of chromosomes move to the cells, move from the cell's equator to the poles. This also happens in mitosis. By the way, prophase 1 is exactly like mitosis pretty much. But in anaphase 2, our centromeres divide and our chromatids move to opposite poles. Notice that we have now chromatids, and then we no longer have these homologous pairs of chromosomes, because you don't need a pair yet, because you get one from your mom and one from your dad. So that's how you get your pair. And then we have telophase 1, and this is where the chromosomes will gather at the poles, cytoplasm starts to divide, we go through cytokinesis, and let's look at telophase 2. In telophase 2, the nuclear envelope reforms. That's a little bit different because we don't have a nuclear envelope that breaks down in prophase 2 because it was never reformed at telophase 1. And so that nuclear envelope being able to form around each set of chromosomes, that's really important. Also, at the end of telophase 1, we'll go through cytokinesis and you have 2 cells and they are still diploid. But at the end of telophase 2, that nuclear envelope reforms and now we have 4 cells that are haploid, or half the genetic information. And those cells are gametes. So let's compare mitosis to meiosis. So mitosis is making new cells that are used during growth, repair, development, and asexual reproduction. By the way, in asexual reproduction, binary fission is the bacteria's way of going through mitosis. And for yeast, it would be budding. They produce genetically identical diploid cells. They are exactly the same as the parent cell. The 2 daughter cells are genetically 100% the exact same thing. However, in meiosis, we have cells that are able to reproduce sexually, and they'll have reproductive structures, such as for males, we produce sperm, which are genetically different than egg and look different than egg. But let's see what that looks like when we start comparing them. Okay, so mitosis versus meiosis. Attack of the clones versus dare to be different. So we are counting down the chromosomes, but let's restart this animation. So we start with 1 cell, goes to 2 cells genetically identical. Dare to be different. They split again, and now we have 4 cells. And notice all 4 cells are genetically different. In mitosis, 46 chromosomes throughout. In meiosis, we start with 46, end with 23, because you're getting your second half from somebody else, aka half from mom, half from dad. This is also why siblings don't look the same. So how do we keep up this genetic variation? Well, genetic variation is made through sexual reproduction. Again, we are not going to talk about the birds and the bees, but we're going to talk about the gametes that got you here. So we have genetic variation. This is done in several major ways: crossing over, independent assortment, and random fertilization. So let's talk about crossing over first. So during prophase 1, our homologous chromosomes are lined up next to each other. Notice they're going to get ready to go into metaphase 1, where they're going to line up all on the equator, pairing up. Look what's happening here. Paternal and maternal. So these are all lined up here. And now all of a sudden a switcheroonie occurs. And at the bottom here, we are seeing that half of your chromosome is no longer that chromosome. It is something else. It is crossed over. So we have that mixing of the orange and red crossing over. That's genetically different. So those sister chromatids are actually exchanged with another chromosome. So now you no longer have the same genetic information on there. That happens naturally in prophase 1. When it says paternal and maternal, we're not talking about we're talking about your chromosomes. Half of them are from your mom, which is maternal, and half of them from your dad, which is paternal. So when your chromosome number 1 and chromosome number 1 come together and they switch over, your offspring can have a mixture of a paternal and maternal chromosome number 1, not you. Then there's independent assortment. So in metaphase, we all line up across the poles, and then your pairs or chromosomes are separated out. And then from there, we line up again in metaphase 2, and then we separate them out one more time. So they're independently assorted or random distribution. You don't always have your paternal chromosome number 1 going to the left and your maternal chromosome number 1 going to the right every time. They split up and it's randomly. So you might have them in a mixed up order. It goes right one time, left the next time. So they're not always going to be all your maternal are going to be on the right side and all your paternal will be on the left side. There's a mixture. And so that becomes kind of fun when you think about this. They're independently assorted and they never repeat the combination, which is super cool for that. And then we have what we call random fertilization. It's just that, random fertilization. So this is my birds and bee talk for you. Random fertilization happens because one egg is released every 28 days-ish in females. This one egg is leaving the ovary and going down the fallopian tube on its journey to the uterus. But if something happens, there's a 3-day window, by the way, roughly, where that female would come in contact with a male and their sperm would be released and enters into the uterus. It will find its way up a fallopian tube. Remember, one egg, 2 fallopian tubes. 50% of the sperm don't even find the egg. They just perish. And then if you're the lucky sperm, so then it's a race. Who's going to get to the egg first? You find the fallopian tube, you get in there, and if you win, you'll be the only sperm, hopefully, to fertilize that egg. And so it's random. There's millions and millions of sperm that are released, while there's only one egg that's released. So if you think about this, there's like 70 trillion possible combinations that can occur. And if you think about this, how many of you have a sibling that you look exactly like, and how many of you have a sibling that you look nothing alike? I'll use me as an example. I'm 6 foot tall. My little sister, who's my Irish twin, 15 months to the day younger than me, is like 6 inches shorter than I am. She does not look like me. We have completely different facial structures. We don't have the same nose. We don't have the same eye color. We have the same mannerisms, like the way we talk and act, but that's environmental. We could be put in a room and you would never know which one is which. And that's okay, because it's cool to be a little bit different than your siblings. And so that's random fertilization, which allows for genetic diversity. Again, random fertilization, independent assortment, and crossing over, the 3 things that lead to the genetic diversity of organisms. And we're going to talk next on the multicellular life cycle, but not in this episode. Thank you.